Okay. We were doing photosynthesis, and we still are. That's what we're doing, is photosynthesis. Okay, so what we had done is taken a chloroplast out of a plant or an algae cell, and we diagrammed the plant cell, and within that we had a chloroplast, and we diagrammed the chloroplast and labeled all the parts. And we had a thylakoid within that chloroplast, and I'll just draw a chloroplast. So this is it, chloroplast. Double envelope on that, and then we had these stacks of membranous structures, which we call thylakoid. An entire stack of thylakoids was called a granum stack, and then the fluid we had out here was called the stroma. So what I did was just say, okay, we're just going to take a chunk of one of these thylakoids and we're going to enlarge it. Okay, so that's what I'm drawing here. That's just a piece of a thylakoid. It's made it really, really thick. So this is just a thylakoid membrane. Okay, and we had two pigment complexes. We had a small pigment complex, and then we have a really large pigment complex. And the smaller one they call photosystem 1. And the only reason they do that is because it was the first complex pigments that they discovered. You'd think they would have discovered the big one first, but they didn't because it's larger. But they found the small one first. Photosystem 1. And this one has a chlorophyll in it, which is called a P700. So that is a specific type of chlorophyll. So this is chlorophyll P700. The longest wavelength that it can possibly absorb would be a wavelength of 700 nanometers. Now that's a red light is really what it is, longest wavelength. Okay, and then this one, which of course it has a different chlorophyll. This one is a chlorophyll and they call it P680. That looks like page 680. I'll take that period off of that. Shouldn't have it anyway. Okay, so anyway, this is another pigment complex, just a bunch of different pigments. It has reds and it has blues and it has yellows and all kinds of different pigments, just all grouped together. That's why they call it a pigment complex. So this is photosystem. Yes, ma'am? Oh, I did? Well, I must have been on medication. Because this one is actually, in photosystem 2, you have the P680. In photosystem 1, you have the P700. That was just my brain flipping around on me. It does that sometimes. Actually, much more often than it used to. So I apologize for that. I apologize for getting older. And then I take that apology back. I just apologize for making mistakes. And I might actually make a few more today. I may totally botch this thing completely. You never know. Could be just a bad, bad day for me. And you could all have a bad day today. I'm hoping not. Okay, but like I said, this you have all kinds of colors in here. You know when we did the chromatogram with the spinach leaf, or one of the groups did a chromatogram. You actually smushed some spinach into a little piece of paper, rectangular, and put it in a solution, and the pigments rose up into that paper, and it separated the pigments by a really mass, how heavy they were. The heaviest ones stayed at the bottom, and the lighter ones went all the way to the top, so forth. So we actually had 4 different bands bands, or possibly even 5 different bands. So we knew that there were several pigments in there. And so that's what you're getting here in these photo systems. So we can put the other pigments in here. I don't know that plants actually have whatever color this would be. I'm not sure what color that is, personally. And I'm not sure whatever color it is the color that I am looking at, because I'm colorblind. To a certain degree, I'm colorblind. Like I'll look at a rug at home and go, what color is that? You're like, well, that's actually green. I'm going, and it looks blue to me. Pretty much colorblind, large degree. So I'm putting in some other pigments. They call these accessory pigments, and also sometimes they'll refer to those as antennae molecules. So this little group that we have right here, from here to here, we'll call those accessory, if I spelled accessory correctly. I think I did. Pigments. Or antennae molecules. I like that one much better, actually, because what they're going to do is they're going to trap different wavelengths of light, and they're going to push those different wavelengths of light to chlorophyll. Chlorophyll is actually where the reaction begins. So you want to send as much light energy as you possibly can to chlorophyll, because that's going to be where you're going to get your energy. Obviously, to make sugar, it's got to come from the sun. So you're going to trap it with other colors, and you're going to push it over here. If you'll remember, the green is going to reflect the green light, and of course the yellow would reflect the yellow light, and so forth. So the more pigments you have in there, the more you can have, the more different wavelengths of light you would actually absorb. So that's really, really good to have different pigments. And again, that comes up, that gives us why they would refer to this as a photosystem. So accessory pigments, or we call this antennae molecules. It's just to pick up as many wavelengths as possible. So yeah, there are several different pigments there. Okay, so, and what we're doing at this point is what we refer to as the non-cyclic. We did the cyclic electron pathway. That's what the cyanobacteria, the blue-green bacteria, do. So they're bacteria. They're pretty simple. They don't have chloroplasts. They just have an infolding of the plasma membrane we call the thylakoid. So this is the non-cyclic. We're not going to recycle electrons. Non-cyclic electron pathway. And this, of course, is another type of light reaction. And this is what is going to occur with your eukaryotic plants, also with your algae. Those are the cells that do that, the algae and also the plant cells do the non-cyclic electron pathway. Cyclic electron pathway occurs with the blue-green bacteria, also called the cyanobacteria. So, you know, keep those two processes separate. This is a little bit more complicated. Already we know we have 2 photosystems, so we've complicated it a little bit more, and 2 different chlorophylls, so that complicates it a little bit more. But we're not going to recycle our electrons. So we need a source of electrons. Well, our source of electrons will be agua, water. This is why you water plants, and we just started to get into this yesterday because most people don't have a clue. They know that if you don't water a plant, it's going to die. That much they do know. You add the water, it's sunlight, you know, gets nutrients out of the ground, absorbs gases out of the air, it puts all that together to make sugar, and it grows, it stays green, can live for a certain length, depending on what the plant is. Have very short life periods. It's like in Odessa, Texas, you know, for back probably during the 60s and 70s before, you know, when your grandparents were somewhere back during that period of time, probably. Not really sure. But they planted fruitless mulberries all over Odessa, fruitless mulberry trees. Well, no one told them, I don't think, a fruitless mulberry tree only lasts for about 25 years. So we still have them in town, but they all look like doo-doo, yes, I would say, probably. They don't look good. They look pretty bad. It's because, you know, they're way past their life expectancy, and they're just kind of hanging on by a thread, and they look pretty awful. So not a good tree to plant. You know, if you're planting trees, I would certainly put in an oak, something that will last hundreds of years, not just 25. And pines can last hundreds of years too, so those are really good. Plus, pines are drought tolerant. Well, they don't grow that deep. You can hear anything. A lot of people say, well, I mean, seriously, and you can read anything. Absolutely. Yeah. I mean, it's all kinds of misinformation floating around out there in the world. But what we want to do is understand what we do with the water when we water the plant. So yes, we water plants. We understand that. And what happens with that water, the water is going to break up. So here's my water. So we have two hydrogens and an oxygen. Well, what I want is I want the electrons out of the hydrogen. Everything on life is hydrogen powered. So I'm going to take, from the hydrogen, I'm going to take two electrons, and I'm going to pass those two electrons to my chlorophyll, which is P680. So two electrons. Well, what that's going to leave, if I take the two electrons off the two hydrogens, if I have two hydrogens, hydrogen is one proton and one electron. And then, of course, I have another hydrogen. I have two, H2O. So I have one proton and another electron. So I just grabbed my two electrons and I passed those over to chlorophyll. That's what I did with them. So I took them off. Well, that leaves me with two protons. I need to do something with those. Well, the two protons out of the hydrogens are going to go in here. Well, this is inside the thylakoid. These are your protons. Or we can call them hydrogen ions. It's exactly the same thing. Hydrogen ions, H+, two of those. All right. So I've completely broken the hydrogen apart. I put my two protons inside the thylakoid. This is inside that back thylakoid. So my hydrogen ions are in here, H+, are in there. Or you can put P+ if you want. That's where we place them. And the electrons went over to chlorophyll, and they're in that membrane, within the thylakoid membrane, into photosystem 2. So it actually starts at photosystem 2. So this is why we're watering plants. We want to put the positive protons in here, and we want to grab the electrons, and they go over to the chlorophyll. And we'll begin moving them around. Okay, what's left over is the oxygen. The oxygen will go out. And that's just one oxygen, so really you'd have to like double this in order to get O2. So one thing that it could actually happen to the oxygen, it could go out of the plant. It's where we get our oxygen, you know, for many, many millions of years there wasn't any free oxygen in the environment. And then we started to have oxygen begin to build up in the environment because we did have the evolution of the plants, the algae and so forth. So they began to release oxygen and it began to build up in the atmosphere. Now all of a sudden we could have other creatures that could use that oxygen in respiration and produce a tremendous amount of ATP. Before that you couldn't have cellular respiration. So you have to have photosynthesis before you could have cellular respiration. You have to have oxygen. It goes into the mitochondrion inside the inner membrane. That's where that went. So two things can happen. This can either go out into the environment, which is where we get our oxygen, many people do you think actually know the oxygen that you breathe, where did that come from? But where did it come from? It comes from the upper part. Get the electrons out of the hydrogens, the protons go over here to the inner thylakoid membrane, electrons go over to the chlorophyll, and the oxygens can go out into the environment. So it actually comes out of water. When you water your plants, the plant is releasing oxygen. It makes an abundance. So A, it can go out of the plant, but at the same time it does B, which is it goes into the mitochondrion for cell respiration. So that's pretty cool. What that tells us then is plants not only produce their own sugar, their own food, they also make their own oxygen. We have to eat food and we also have to breathe the oxygen. They don't. They make their own oxygen. In fact, they make so much, they make an abundance that a lot of it goes out of the plant. They make plenty to go in the mitochondrion where they can use it to do cell respiration, just like you and I do. Take sugar, break it down, get a lot of ATP, run it through the electron transport system of the mitochondria and generate the ATP. So you do two things with that oxygen. Either it goes to the mitochondria, and anything that's left over that's in abundance goes out of the plant, and then therefore the animals get to breathe the oxygen, so that's actually very beneficial. So we should all just go, thank you, plant, thank you, thank you, thank you. That's really sweet of you to produce so much that we get to have an excess in the environment. Therefore, we can exist because they don't necessarily need us. In fact, at this point, we're producing so much carbon dioxide. Not humans. We're not making very much carbon dioxide, actually, but the automobiles are producing such a huge amount that the plants are just loving it. They're just going, burn that fossil fuel. Burn it, because the CO2 levels are going up, and therefore they're able to produce more oxygen, and they're able to produce more sugar, and they're growing faster and faster than they've ever grown before, and plants are loving this whole global warming thing, as long as they get water. Where the whole system falls down is if they don't get water. If they don't get water, the whole thing falls apart. That's what you have to do. That's the very beginning of the whole process of photosynthesis. Take those hydrogens, pull them apart. Protons go inside the thylakoid, and the electrons go over to chlorophyll. It comes from water. You don't have water, you can't do that. The whole system falls apart. Plants are going to die. So why do we water plants? So we can pull the hydrogen that's in water apart, put the protons in here, and begin moving the electrons. That's why we water plants. And at the same time, we release the oxygen. Now, you're going, hopefully, some of you are thinking, well, what breaks down the water? Two things. One is an energy, which is called light. Light breaks down the water. And the other thing is an enzyme. An enzyme will break down that water. So it's very important to have that enzyme. And they don't typically talk about that enzyme and give it a name. There is one enzyme that they will give us a little bit later, but that will be later on in this process. But at this point, no, we don't really need to know the name of that enzyme. So light has to strike the water to break it apart, plus we have to have an enzyme to break it apart, and then the two hydrogens break apart, and the two protons go in the thylakoid, and the two electrons go over here to chlorophyll. I didn't hear any of that. I have allergies, and one thing that happens when I have allergies, my nose stops up, so I'm constantly doing this and swallowing my snot. Disgusting. I could go and do something else with it. or I could just carry around a handkerchief and just have it all nice and nasty by the end of the day. Or I used to use Kleenex and my nose would look red. Nah. And the other thing that happens is my voice starts to get real, just like close off on me. And then the other thing that begins to happen is my ears close up. And it's not like I'm not hard of hearing already, but then they start stopping up on me, and it's just like, I am stopped up. Ears, nose, throat. It's a wonder. I'm having a hard time breathing. It's horrible. It's called allergies. It happens in the fall because we get a lot of rain, and then all the mold, the fungi that live in the ground, they start producing spores. They just shoot spores into the air, and then I breathe them, and I have an allergy to the spores. If you notice on the newscast, they'll always give you like a pollen count and a spore count. And right now, molds are just like really, really high. Molds are huge, and that's why I have all these problems. But, I managed to get through it. I don't take drugs for that, for allergies. I know I should, but, I suffer through that. Okay, I'm sorry, though. You had a question. Right. Well, the CO2 comes from all kinds of sources, we being one of them. But the cows give off a lot of CO2, and so do the geckos and the snakes and the animals in general. Okay. So far we've just broken apart water. That's really all we've done. Passed electrons to the chlorophyll, which is what we need to do. And we've thrown our positive protons in here, which are the hydrogen ions. Okay, so at this point now, light is going to strike that chlorophyll, and I'm going to you got lines going all over the place. I understand. It's like, man, this is complicated. Look at that. Not really complicated. We just broke water apart and we passed some light over to chlorophyll. That's all we've done. Not like rocket science. Anybody confused yet? Well, it'll go to both, but we'll do that in stages. Okay, so the reason that we absorb light here, and the light that's absorbed by these accessory pigments gets channeled over to the chlorophyll. So we want as much light as we can possibly get to that chlorophyll. And the reason we want to do that is because we want to remove the 2 electrons that we just got from water. And we're going to pass them to a molecule which is called Z. We've seen this before. In fact, this will look very similar. And then we want to start moving this molecule, the electron from molecule to molecule. So we pass those same 2 electrons from Z to this molecule, and then we pass those same 2 electrons from that molecule to this molecule, and then we pass those same 2 electrons from this molecule to this molecule, and then we pass those same 2 electrons from this molecule to that chlorophyll right there, 2 electrons. So what we're doing here, this is an electron transport system. Electron transport system, or you can call it electron transport chain. It is a redox set of reactions. Redox, we've talked about that. Oxidation reduction. Remember, oxidation is you remove electrons. So she grabs the electrons, 2 electrons from me. Okay, she grabbed the electrons from me. All right. I'm oxidized because I lost them. She's reduced because she grabbed 2 electrons. She is more negative. And she passes them back to me. She's oxidized. Now I'm reduced. We can play this longer if you'd want. Okay, so I pass them back to her. I'm oxidized. She's reduced. She can pass them to me. She's oxidized. I'm reduced. I now have more negative charge. Thank you. Thank you. All right, so it's an oxidation reduction reaction. That's all it is. And also, which we've talked about this, we're really taking electrons from a low energy shell, we're passing them to a higher energy shell, and then we're passing them to an even higher energy shell, and then we're passing to an even higher energy shell. And if this were cyclic electron pathway, those two electrons would come right back down to where they started. This is not cyclic electron. Not going from a high energy shell back down to low. That's what you do in cyclic electron pathway. That's how the blue-green bacteria do that, the cyanobacteria. But what we're going to do is we're going to use light to remove those two electrons off of P700, chlorophyll P700, which is in photosystem 1. So that's my light again, light. And those two electrons are going to go out of thylakoid. So this is out of thylakoid. And what that means is we just pass those two electrons out here. Well, what is this? This is stroma. That's a fluid. Okay, so we put our hydrogens in here, H+. We put those in here, and we pass our electrons out into the fluid. So far, that's all we've done. You look at that and you go, really? Looks like you did a whole bunch of stuff. You look at that and you go, it's a lot of stuff happening there. No, we just broke water apart. We threw the electrons over here. Light hit it. They went over here, and then we moved them over here. Light struck that, and then we took the electrons out. Protons from the hydrogens went in. Oxygen went out or went into the mitochondria. That's basically what happened. Okay, so those two electrons we passed out here into this stroma, they're gonna pass to a carrier, and we've seen hydrogen carriers before. If we're doing the cellular respiration, we would say NAD or FAD. This is with plants, so this is a different carrier, and this one is called NADP. So if you think of P as plants, then you're cool. Then you go, okay, I can remember this. The NAD and the FAD, that's for cell respiration, which of course does happen in plants as well as animals. But the NADP is just for photosynthesis, NADP. And the P doesn't stand for plant. It stands for a phosphorylation, or a phosphate group, basically. It's a nicotinamide adenine dinucleotide phosphate. So that's what the P stands for. All right, so we're going to go to an NAD. So now here's my NADP out here, and it picks up those two electrons. I'm going to put a -2 on it. Okay. So how many times do we do this? I don't know. How much water do we have available? Put that water on it, or did you put that much water on it, or did it rain for a week? You know, how much water do you know? Did you guys how to put them in? They're simple. People are like, you know, had sprinklers out and they're going around. I'm going, what? Put in an irrigation system. You know, in this development where I have a house in Alpine, there's this lady, and I'm sure she's like way up in her 70s. I don't know how old she is. She could be in her 80s for all I know. But I know she's had a lot of chemo, and I'm not sure whether it was breast cancer or what. And there for a while, I saw her out in her yard. Now, her yard is like on a hill, and she can't walk. You know, she's just like doing this. She's like walking down the hill with a hose, watering all of her plants. And I'm going to irrigate all your plants for you. She goes, what? I go, I'm going to put in some irrigation so you don't have to walk around here trying to water all these plants. You know, you're going to do what? You know, and in like 4 hours, I mean, everything was irrigated. Everything had a little bubblers on them that pop up, and I'd have a timer on it, you know, so this section would come on for this, and that section would come on for that, and she was like, so sweet, so thankful. I'm like, what? It's 4 hours of my time. It was nothing, you know, and the equipment didn't cost anything anyway. Seriously, it's just so cheap, and it didn't take any time. Anyway, so wasn't anything to it, but she loves it. Absolutely adores her irrigation system. So yeah, you know, you put a whole bunch of water on this stuff. So you can do this thing over and over and over. You put a whole bunch of water on here and you keep slamming all inside your thylakoid. You keep throwing them in here, and you keep throwing all your electrons on NADP, making them negative. So say we've got times thousands of these, I don't know, lots and lots of those. We say thousands of these, I don't know, we got lots and lots of those. So you know where we're headed. You have built up a really, really, really huge, strong positive charge on this side of the membrane, which are all these positive ions, which we call hydrogen ions. And you built up a really, really negative charge outside this membrane, which are these NADP minus 2s out here. That's where the electrons end up. And then you have this wonderful little enzyme, which we call ATP synthase. And of course, what we're going to do is we're going to take our NADP minus 2, and we're going to take our positive charged particles, which are H pluses or protons, same thing. But I'm going to write H pluses, so I'm going to put 2 H pluses here, and it's going to pull those particles together. Okay, that's an H now. All right, so this is going to go this way, which is the positive particle. This is going to come this way, which is the negative particle, and they're going to slam together. And if I slam a positive and a negative particle together, what do I get out of it? Who said that? Bless you, my child. Thank you. You made my life worth living again. One more time. I'd almost given completely up. You know, I was almost down to the razor. Yeah. Almost down to the razor. You know, you have kept me going. I want you to know. That's super. Wait. So energy is going to come out of that when I slam my positive and my negative together. And what that's going to do is give me enough energy so that I can take ADP, you know where I'm headed, P plus my phosphate, PI, come on, work you, pen, and I take ATP. All right, that's one product right there of the non-cyclic electron. Is if I get NADP minus 2, my bonds together. The other thing that's coming out of this is going to be NADPH2. Right. So I've actually produced two products right there. So those are the two products, the non-cyclic, and that's how that works. So real quickly, I'll run through it. This is the Little Red Riding Hood story. To me, they're all stories. That's all they are. It's all a story. We all know how to tell the Little Red Riding Hood story, or the Wolf and the Three Bears story, or, you know, we remember these stories from our childhood. Although my mother never actually told me any of them, best I can recall. And I've never confronted her with that. I go to dinner with her typically on Tuesday nights. Tonight might be the night. I just go, why didn't you read me stories when I was a kid? Little itsy bitsy spider. I didn't get that. I had to learn those from other people. Other people had to tell me. Well, didn't you learn the itsy bitsy spider? I'm in I'm like in grade school. I'm going, I don't know what it is. I know what a spider is, but what are you talking about? I have to learn the itsy bitsy spider thing. Mom was on that. I know. We should let them become friends. Two mean women, who needs to tell a kid some stupid thing like that? Well, it's part of growing up. Anyway, so we'll go through this thing again. All right, so we have to begin with water. We've got our two photosystems, blah and blah. We need some light. One thing we're going to do is bust up water. That needs light and an enzyme to do it. And, of course, from the water, what we want are the hydrogens. Everything on Earth is hydrogen powered. All right. That's Indian. That's my Indian lingo there. All right. It's the best I can do. All right. All right. So I can do a really good Spanish accent, though. That one is good. Okay. At some point. I'll wait. I'll bust it out. You just wait. Okay. So I'm going to take the two hydrogens. I can barely do it in English, the whole Texan thing, I go to New York and they go, you're from Texas, right? And I go, you could tell. I go, yeah, talk some more. They just want you to talk and talk and talk. Because they just like, shouldn't you have on boots and a hat? It's like, you've got such a Texas accent, you just scream Texas. It's just like, man, is it really that noticeable? They're like, yeah. It's funny. Anyway, okay. So we're going to break water apart, but we're really going to break the hydrogen apart is what we're really after because we want to get the protons and the electrons, as we mentioned right here, protons and electrons. So we're going to take our protons, we're going to throw them in the thylakoid, we're going to take our electrons and we're going to move them over to the chlorophyll. The oxygen is going to either go out or the algae, or it's going to go into the mitochondrion, which is going to have oxygen in there. Okay, so then the electrons are going to go off the chlorophyll because light strikes the chlorophyll, and that removes the electrons. They go to carrier molecule Z, and then they go to these other molecules. We go from Z to one molecule to another molecule to another molecule. Finally, we go to photosystem 1. Light strikes that, and the electrons go out of the thylakoid, and they land on NADP, making it negative. Keep in mind, we now have our hydrogens inside here. So we've got lots of water, hopefully, because in this part of the world we irrigate, typically. And so eventually you make a whole bunch of negative NADPs out in the stroma, the fluid, outside the thylakoid, and a whole bunch of positive hydrogens, or protons, inside the thylakoid. And then this enzyme allows them to come together, the negative and the positive. And as soon as together, you get enough energy to make ATP. And of course, if you get your electrons that are on NADP back together with your protons inside the thylakoid, you're going to regenerate hydrogen. So now you have NADPH2. That's not a cyclic electron pathway. How it works. This is why we water plants, and that's how we get our oxygen. And for people to go, I don't care to know how we water plants, and I really don't care where our oxygen comes from. I'm like, really? You don't really care? I mean, I don't understand that. Other than I do, because I don't know what a carburetor is. I really have no clue. I don't know. My automobile's in the shop right now because the oxygen sensors need to be replaced. I'm like, what? It's got an oxygen sensor? What's it doing in there? I'll stop breathing while I'm in the car. I'm sucking too much oxygen out of the interior. I don't know. Automobile's in the shop. Stop breathing. Okay, now, the very last thing that we need to do is we need to look at, and if you'll remember, when we first started this whole whole chapter. We said that we had photosynthesis, and we've divided that out into two major reactions. We had the light-dependent, which we just talked about, light-dependent reactions, and we divided that out, and so we did the cyclic electron pathway, which that would be the blue-green bacteria, the cyanobacteria. They just recycle electrons, basically put them on higher energy shells and pull them right back down to where they started in chlorophyll, generate some ATP. And then we do this non-cyclic, which we did just now, non-cyclic electron pathway, and we generated ATP, and we also generated NADPH2. Those were your two products. Okay. And then the second part of this, we need to look at what is called the light-independent reaction, and it's really pretty simple. And it has light independent. It can happen in the daytime or the night. It just doesn't need light for this process to work. Who does it? What cells do this? Plants and algae again. Where does it happen? It happens in the stroma, the fluid around the thylakoids. Stroma, that's where it happens. So then we need to kind of look at what we start with and what we end up with in that process. So light independent. See what we got. We're light independent. Okay, so we're going to imagine this is a big, huge chloroplast. These are our thylakoids right there. And what I have just done is I've just pushed out ATP, and I've also pushed out NADPH2. Okay, that was from the non-cyclic electron pathway. So we're out here in the fluid. This is the stroma. All right. First thing we need to do is we need to get some carbon dioxide at the end of this. Where does the CO2 come from? Well, you know, all animals exhale CO2, carbon dioxide. Your automobile gives off a lot of CO2. All the machines run on gasoline and so forth are giving off a lot. Plants are happy. Is warming up significantly fast, and that can be a problem for everybody. Climate change. Pretty funky out there for sure. Crazy. All right. All right. So we're not what, going to be degrees outside today? We should be in like, cutoffs and, that's all. Flip-flops. Well, yeah. Y'all could wear Band-Aids if you're female. Work. I'd vote it in. Yay on the Band-Aid thing. Yay. Yay. Vote. So we're going to get in some carbon dioxide, CO2. Where does it come from? Well, you exhale it. It goes into the leaf. It goes into the plant. It goes into the stroma, CO2. It's going to combine with a 5-carbon molecule. That's not just 5 carbons. It's got other things attached to it. But it has a name, RUBP. That stands for ribulose bisphosphate. OSE, bis means you have two. So it's not all that complicated. It's just a sugar with a couple of phosphates on it, ribulose bisphosphate. Okay, so the 1 carbon, CO2, combines to the 5 carbon, and that gives us a 6 carbon. This is unstable. It's not going to stay that way. It rapidly breaks into 2 3-carbon molecules very quickly. So a 3 carbon here and a 3 carbon here. Those 2 3 carbons are called PGA. They have a name, PGA. Right? PGA is phosphoglycerate. And the PGA, that's the very first stable compound in this reaction series. So sometimes they call this whole process right here the C3 pathway. Because the very first stable compound is a 3-carbon molecule, PGA. That's why it's called C3 pathway. The gentleman who discovered the pathway, which was his name, believe it or not, was his last name was Calvin. Now, his last name was Calvin. Would you name your child Melvin? I think not. His parents did. They did. They go, we're going to call him Melvin, and his name will be Melvin Calvin. You know, teacher calling row, Melvin Calvin. That's got to be a typo. Marvin Calvin. He goes, it's Melvin. it's Melvin. Yeah. They stuck me with that. Actually, there were 2 gentlemen that discovered this process. The other one was Andrew Benson, and they kicked him completely out of the biology books. I don't know what he did, and to get kicked out, probably didn't do anything. But you know, I think if I were the Andrew Benson descendants, I'd be a little bit upset about that. Like, you know, Granddad came up with this too. You just took his name completely out. Who are you to take his name completely out of this deal? You know, great Granddad Benson, Andrew. No, remember that? He's not in. Anyway, they just Calvin, Melvin Calvin. Anyway, sometimes called the C3 pathway, also referred to as the Calvin cycle after Melvin. All right, C3 pathway. Okay, at this point, we're almost finished. We're almost there to make sugar. We're out here in the fluid stroma. All we've got to do at this point is take that 3-carbon molecule, and we made 2 things, the ATP and the NADP, H2. Okay, and I'm going to use those right here. It's going to take, I want to remove my hydrogens off of NADPH2. I'm going to take those hydrogens off, which will just leave me with just NADP. So I'm going to do that on both sides. I'm going to take hydrogens off of NADPH2. And that just leaves me with NADP. But in order to do that, that requires some energy. I have to take my ATP and break it down into ADP and PI. So ATP, I pull the last phosphate off, and it becomes ADP plus the 3rd phosphate. And that provides the energy to take those 2 hydrogens and put them on that 3-carbon. So one more time, in order to do this, remove the hydrogens, I'm going to take ATP, I'm going to break that 3rd phosphate off. That leaves me with ADP plus PI. That's a PI. And we released energy. So what I've done now is I have taken these 2 hydrogens right here, those two, that originally came from water. That's where we got them, from water. And I'm going to put them on a 3-carbon, 2 hydrogens that it didn't have before. because I just took them off NADP and I put them on that 3 carbon, which gave me a 3 carbon with 2 hydrogens. Okay, so I do the same thing here. So now a 3 carbon. Well, these of course have a name as well. They're called PGAL. This one's also called PGAL. PGAL has a name that stands for, I know you love these, phosphoglyceraldehyde. Again, do not even think about playing Scrabble with me. I can make those letters into all kinds of biological words. I don't know what it is, but I'm sure he's right. You know, I play Scrabble with people now, just biology words, and they'll go, well, looks right. You want to Google it? They go, no, just give him credit. And I just totally made it up. Messing with them. Just messing with them. Like, I win. Man. Anyway, the point is, on these PGALs, any 2 PGALs can be made into sugar. So any 2, here we're just at the final hurrah here. Any, bad pen. PGALs can become, we'll call it glucose. Okay, we made sugar. That's what we wanted to do. Synthesis right there. We wanted to make sugar. You put any 2 of these together. How many times do you do this reaction? This reaction happens thousands and thousands and thousands and thousands of times, depending on how much water we have, depending on whether it's cloudy for a day, how much sunlight we have. This depends. Okay. Other PGALs can be used to make this thing right here. Okay, right there. So other, since we're making thousands of them, other PGALs used to remake RUBP. Okay, so I've made my sugar. Here it is. It's what I wanted to do. It's called photosynthesis. And I've had to make this stuff. Got to have it. The whole process isn't going to work if you don't have RUBP. And the enzyme used to put the CO2 in the 5 carbon, there's an enzyme there. It's called Rubisco. And that's a very important enzyme because that is the most abundant enzyme on planet Earth. The most abundant enzyme on the planet. That makes it something you ought to know about. Because if you want to get into med school or vet school or dental school or pharmacy school or something like that, I promise you on that entrance exam, Rubisco is going to be on it. It's the most abundant enzyme on the planet. It's when you take the 1 carbon, combine it to the 5 carbon to give you a 6 carbon that's unstable that immediately splits into 2,3 carbons, which is why they call it the C3 pathway. Calvin Benson discovered the process, and then you throw a couple of hydrogens on there using ATP. You make PGAL. Do it over and over and over. Make sugar, make more RUBPs. Shazam! You did some photosynthesis. So you got to do that light reaction, non-cyclic electron pathway, and then you have to do the light-independent reaction, which is this thing right here, C3 path, and that happens out in the stroma. Okay, we're not quite finished with this chapter. There's just a itsy-bitsy spider we got to get a hold of and finish this thing up with, but we are out of time. In fact, I used 5 minutes of your time, so I'm going to give you 15 whole minutes to run and play and do what you want to, and we'll see you in the laboratory.